right, everyone, if you could find your seats, please. We're going to start the next session. And I also wanted to say to the parents, you all had a lot to share, and it's moving to hear your stories. I want to remind you that that's the value of Matrix. You know, share your stories there. Right now, you're each individual voices, but by completing the surveys in Matrix and sharing your data with Citizen, you go from just being one individual voice to being one of many. You know, we have 190 people who are part of Cure GABA-A, and so to the parents, share your data there, and also then we can give it to all of the researchers instead of just those who are here with us today. So please know that that's the most valuable place for you to be sharing your stories. So with that, we're going to now move on to a little bit more of the basic science side of things. We were going to have a talk from Dr. Stephen Moss, but he was not able to be here today. So instead, we have Paul Davies from Steve's lab. And so Paul has his own talk on his own work, as well as will be sharing a little bit of the other stuff from Steve's lab.
Yeah, there you go. Probably false advertising to talk about, I'm going to talk about targeting human GABA responses. That's the ultimate aim in the lab, that we would love to talk about, look into human GABA receptors and human GABA variants. What I am going to talk about, though, is ion channel subunit variability and some of our work involving neurosteroid modulation of GABA receptors. So, there we go. So, when I think about how we're going to treat people and modulate GABA receptors, there's a couple of questions that always come to my mind, and such as we need to know where GABA receptor subunits are expressed, both within the cell and also within circuits within the brain. We need to understand how neuronal activity affects expression of GABA receptors. And my talk yesterday, I showed some early work following seizures. There's a change in subunit expression where there's an increase in delta and epsilon subunits following seizures, and we really don't fully understand why or how and the consequences of such changes. We also need to know how drug exposure affects expression of GABA subunits, and obviously with this group we need to understand how variants within subunits affect their transport and their expression and their function. So this is a typical slide that you see all the time. The GABAergic presynaptic neuron releasing GABA into the synapse and activating synaptic receptors and extrasynaptic receptors. And if you look into the literature, it's always seems to be quoted that synaptic receptors are made of alpha-1 beta-gamma, alpha-2 beta-gamma, and alpha-3 beta-gamma, and extrasynaptic receptors are either alpha-5 beta-gamma or alpha-4 beta-delta or alpha-6 beta-delta. The main question is, like, how does the neuron transport these different subunits into their locations within the synapse or their extrasynaptic sites? We've done proteomic data that doesn't agree with this common separation of subunits. And I pose the question that is the current dogma preventing us from developing new, novel GABAergic compounds? And we've worked a lot with a lot of drug companies and also looked at drug company websites, and there's lots of effort and money spent on developing compounds that target these specific subunit combinations. But if those subunit combinations don't exist physiologically, then we are wasting time, money, and effort developing such compounds, leading to drug failures when they go into clinical trials. So what do we know about how synaptic and extrasynaptic receptors are trafficked to their precise locations? And Steve Moss has spent a career looking at how phosphorylation changes to GABA receptor subunits affect their individual trafficking. So GABA receptors, the subunits are formed together in the ER, and if they become transport ready, then they are trafficked up to the plasma membrane and inserted into the plasma membrane. And even synaptic proteins are inserted into extrasynaptic sites, and then they diffuse into the synapse where the alpha subunits bind with high affinity to anchoring proteins at synaptic sites. And the extrasynaptic receptors, they don't have any signaling for those anchoring proteins, and so they just diffuse within the membrane in the extrasynaptic space. So protein phosphorylation greatly affects the rates of insertion and endocytosis of the GABA receptors subunits. And so we've spent a lot of time looking at phosphorylation of gamma subunits that stabilize gamma-containing receptors within the membrane at synaptic sites. Beta-3, we've spent a lot of time looking at two residues, S409 and S408, and when they are phosphorylated, it stabilizes those receptors within the membrane. When they become dephosphorylated, these residues bind to AP2 and the receptor is endocytosed. And it can be repackaged or it can be degraded. In the extrasynaptic space, alpha-4 has a serine at 443, and when that's phosphorylated, that increases the insertion rate of that subunit. Beta-3, again, forms with alpha-4s, and again, phosphorylation of 48, 49 stabilizes in the membrane. So the overall picture certainly for extrasynaptic receptors, which I've spent a lot of my time on, phosphorylation increases insertion and decreases endocytosis, leading to greater accumulation of extrasynaptic receptors. Now, sometimes when I talk about this, I get looks of amazement from GABA researchers. When I say GABA receptors are not static proteins in the membrane, they're constantly being turned over. And the recycling turnover happens about every 10 minutes, very, very rapidly. And to me, that suggests just how important GABA receptors are for the functioning of neurons. Because if something was less important, you would imagine it would be stable for a lot longer period of time. Whereas if you've got a rapid turnover rate of the receptor, then that gives the neuron plenty of opportunity to adjust its receptor composition minute by minute as the neuron has changes in input and activity. So it can respond to different inputs, and then it can come back again, back to baseline. So we've, in the lab, we've done a lot of proteomic work. It was led by my colleague, Dr. Joshua Smalley, and most of the work was done by a MD PhD student, Abby Lemons, and former graduate student, Catherine Cho. And you can see how long they've been in academia based on the level of their smiles. So we started off looking at synaptic receptors containing alpha-1 and alpha-2. Alpha-1 is a mainly located at synaptic sites on the dendrites, whereas alpha-2 is mainly found at synaptic sites on the AIS site. So two variant sites that we were interested in. And of course, we're interested in alpha-4 as it relates to tonic inhibition. And again, that's tonic inhibition is greatly related to sensitivity to neurosteroids, which we have a lot of interest in. And so we wanted to look at these types of receptors. So Josh developed a process where he centrifuges purified forebrain, and so he can isolate plasma membranes, he can isolate membranes from the ER, and so combining immunoprecipitate with very specific antibodies, and then resolving it on native, a blue native page, and immunoblotting in non-denaturing conditions. So we're not pulling apart the subunits, we're bringing the intact receptor protein along with proteins that are associated with them within the membrane. And then we send them off to for liquid chromatography and mass spec. So this is some of the data that we got. Alpha-2 antibodies aren't very good, so we made this mouse that was tagged with GFP and also MIC tag. And so the MIC tag antibodies and the GFP antibodies are very high affinity. So we can isolate alpha-2 containing receptors from the plasma membrane of the forebrain. Each data point represent one experiment which contains 7 individual mice, and the stars above the bars represent those proteins that were significantly detected above IgG levels, controls. So what was surprising when we analyzed the data was that we could not find receptors that contained just alpha-2. It seemed that all alpha-2 containing receptors also contained alpha-1. They also contained beta-1 and beta-2. Beta-2 and beta-3 and gamma-2. When we repeated the same process but looked at alpha-1 containing receptors, we mainly saw alpha-1 forming by itself. Occasionally we do detect lower levels of alpha-2 and alpha-3 coming down with alpha-1, but mainly alpha-1's on its own. And alpha-1 comes down with beta-1, beta-2, beta-3, delta, and gamma-2. Again, that was kind of surprising to us. Just recently, there was a paper published in Nature where they made a mouse model where they joined alpha-1 subunit with a Fab protein, so an antibody that they could use to isolate alpha-1 containing receptors. And they did mass spec, but they also looked at cryo-EM as well on these receptors. And so what they found is that most of the cells do contain just alpha-1 by itself. So there's 2 alpha-1s, 2 betas and 1 gamma. But they also found this population where they had alpha-1 in combination with even alpha-2 or alpha-3, betas and gammas. And these two populations are different from one another in terms of the position at which the alpha is next to the gamma. So alpha-gamma pockets is where benzodiazepines bind to. So it's very important for pharmacology. And so these two populations, one is the alpha-1 is next to the gamma, and in this population, alpha-2 is next to the gamma. They also pointed out in the paper, which I found interesting, is that in these combinations where alpha-1 was the sole alpha subunit in the receptor, that the alpha-1 subunit was compacted, and the protein shape was not the same as receptors that just had alpha-1 within it. They didn't expand on that. I just thought that was really interesting observation. And it begs the question, what does GABA alpha-1 variance do to the structure of these receptors? And does it change the composition of alpha subunits and beta subunits? I think that would be pretty interesting to have a look at in the future. Okay, so then we repeated this experiment, but this time we compared alpha-1 to alpha-4, so another round of experiments, another round of lots of mice. And so, like the previous experiment, we found that when we isolated alpha-1 containing receptors, they came down with small amounts of alpha-2 and alpha-3, betas 1, 2, 3, and gamma-2. We also detected some alpha-3 and delta. What was surprising to us was that when we repeated this with the alpha-4, we only detected alpha-4 and beta-3 subunits. No alpha-4 beta-delta in these baseline experiments. So the alpha-1 beta-delta, we was initially surprised, but then of course we remembered that Istvan Modi's lab, amongst others, have talked about the gin and tonic current, in that alpha-1 beta-delta forms these extrasynaptic receptors on interneurons that are sensitive to alcohol. So so you've got a principal neuron, and you've got the interneurons feeding onto it, and they have alpha-1 delta. And alpha-1 delta, because of the chloride concentration in the terminals of interneurons, they can be depolarizing as chloride leaves the cell. And this interesting work that was published in 2014 suggested that the level of tonic inhibition in these interneurons determines their firing rate and obviously release of GABA. So at low tonic currents, you have excitation as chloride leaves the inside of a cell and depolarizes the membrane, so you get increased firing of interneurons, so increased release of GABA. But then at high conductance, then you basically short out the voltage of a membrane, and it causes inhibition of GABA release. So you have this biphasic response of interneurons based on the conductance of alpha-1 beta-delta containing receptors, which is something that we have to think about when we talk in terms of gain of function or loss of function of alpha-1 or delta containing receptors, how they are impacting the function of interneurons. All right, so summarizing the subunit variability part of my talk, GABA receptors are not static membrane proteins. Subunits need to be in the right place at the right time in order to respond to stimulus. Phosphorylation of subunits is very important for the stability of GABA receptors in the plasma membrane, controls insertion, endocytosis. I didn't show any figures of this, but some of our data that is published just recently suggests that the phosphorylated state of beta-3 at S408, 409 is higher in alpha-1 containing receptors. And so when the beta-3 is phosphorylated, there's a tendency for those beta-3 containing receptors to go to the synapse rather than to the extrasynaptic site. And GABA receptors may contain multiple alpha subunit types. Now I want to transition somewhat to neurosteroid modulation of receptors. Neurosteroids have had a long history of therapeutic potential. They've always been proposed to have therapeutic potential for seizures, anxiety, depression, pain, neuroinflammation. but only 2 neurosteroids are currently in the clinic. One is ganaxolone, that was approved by the FDA last year for seizures associated with CDKL5 deficient disorder, and that was by Marinus. And Sage has zuranolone for postpartum depression. So neurosteroids are manufactured within the brain from cholesterol, where the mitochondria have the enzymes to convert cholesterol into progesterone, and then progesterone's then metabolized into allopregnanolone. And allopregnanolone is a neurosteroid that binds and modulates GABA receptors. Obviously, women have more progesterone within their peripheral tissues, but within the brains, both men and women have the right enzymes to produce allopregnanolone. Now, allopregnanolone can be induced by stress, and some conditions have deficits in cholesterol, such as Fragile X. And Fragile X, we've looked at allopregnanolone levels within Fragile X brains, and even though their plasma levels are normal, their CNS levels are greatly reduced. So the neurosteroids then are released, and they can bind to synaptic and extrasynaptic receptors where they can potentiate those responses. So it's seen a typical synaptic GABA response that gets potentiated by neurosteroids, so it's prolonged. Longer, there's more inhibition that happens. And similar with tonic currents in the extrasynaptic region, steroids bind, keeps the channels open for longer, and there's more current that's being passed. So how do allopregnanolone and other neurosteroids modulate GABA receptors? So there's an allosteric site. So again, this is the study from Sun, where they, in their cryo-EM, whether they applied allopregnanolone to their PrEP or not, they always found allopregnanolone bound to its binding site, suggesting that there's this endogenous level of progesterone that's always there in the brain, and always modulating to some degree GABA receptors. So GABA binds between the alpha and beta subunit, causes the channel to open, chloride flows through, steroid comes along and binds in this binding pocket between the TM3 of the beta subunit and TM1 of the alpha subunit, and causes this anti-clockwise conformational change in the protein to keep the channel open for longer. The structural change also locks GABA into its activating site. So the channel's open for longer, passes more current, more inhibition. So we were fascinated by these stories that were in the literature about how phosphorylation can change neurosteroid properties in the brain. And so we've been working on this since 2008, and we've seen how phosphorylation can, how neurosteroids change the phosphorylated state of certain subunits, specifically alpha-4 and beta-3 subunits, and how that increases the trafficking. So to get a functional experiment going where we can look into this, we make a brain slice, have it recover for an hour, put it into a separate neurosteroid chamber for 15 minutes, then transfer it to our recording chamber on our microscopes that doesn't have any neurosteroids in or hasn't been used for neurosteroid research, and we wash for at least 30 minutes. And then we try and find a neuron to patch and go whole cell. So by the time that has happened, about 50 minutes have passed where we've been trying to wash out as much neurosteroid from the slices as possible. We have a dual Perfusion chamber, so we're perfusing the brain slice from the top and below. The slice go whole cell, we wait for 10 minutes, and then we apply picrotoxin, which is a GABA channel blocker. Because tonic current, you can't have a readout because it's on all the time, so the only time we can measure that is by blocking the current. So these are some of the recordings where we've got the baseline, put picrotoxin on, extrasynaptic receptors close, and so we measure the difference between holding currents and in the presence of picrotoxin. Those slices that were treated for 15 minutes with allopregnanolone, there's a significant increase in the amount of tonic current. We can co-treat the steroid with a PKC inhibitor, and we can reduce the amount of enhancement. We've been working a lot with Sage Therapeutics with their steroid compounds, and we have this model neurosteroid called Sage 516, and that mimics the effects of allopregnanolone treatment. It enhances tonic currents, and it's sensitive to block by PKC. And we've done the biochemistry to back this up. Both Allo and Sage 516 increases the phosphorylation of S40849 of a beta-3 subunit, and measuring the levels of beta-3 on the membrane surface of the neurons, we also see Allo and Sage increasing the GABA receptors. So how are neurosteroids achieving this change in phosphorylation, trafficking, and increase in inhibition? Long story short, we've published this before, but we believe that neurosteroids are not only allosterically enhancing GABA receptors that are already on the membrane, but they are also binding to these G protein coupled receptors called membrane progesterone receptors. And they signal phosphorylation changes that increase enhancement and decrease endocytosis, leading to an increase in tonic current. So the one thing that we're trying to wrap our heads around is that these therapeutic areas of neurosteroids, and the anxiolytic, the antidepressive, and the anti-epileptic effects, how are those being achieved? Are they being achieved by the allosteric mechanism, or are they being achieved by the metrotropic action, or combination of the two? And that's ongoing research. So what are membrane progesterone receptors? Because there's not much known about membrane progesterone receptors. They're a family of G protein coupled receptors. There's NPR alpha, beta, gamma, delta, and epsilon. And the alpha, beta, and gamma are associated with GI, G protein coupled receptors. NPR delta is GQ, and epsilon is GS. And so we have been producing stable cell lines that express these receptors to look at the signaling cascades and treating them with different neurosteroids to see which neurosteroids activate which NPR and lead to the signaling. So with this Sage 516 that I showed you before, through these cell lines, we found that it's very selective for NPR delta, which is highly expressed within the brain. Incidentally, when you look at the expression profile, it looks as if they're expressed in the same region of the brain as GABA alpha 4 subunits. We also use this other compound that we called ORG. It's a non-selective NPR agonist. It activates all of them. And so, anyway, so both Sage 516 and ORG activate NPR delta, causes an increase in PKC, and then that affects the trafficking of receptors to extrasynaptic sites and affects also the contribution of receptors in the synaptic area as well. So I showed you this earlier where we put on 516 and it increases the tonic current and it's sensitive to PKC. So we do have NPR delta knockout mice. So we repeated this experiment in the knockout mice. So you can see here in the wild type vehicle, that's the level of tonic currents, 15 minutes of 516, followed by at least 15 minutes of wash. Then we see this increase in tonic current. But in the knockout mouse, we do not see 516 affecting the tonic current. There's no increase. So I mentioned before that Sage 516 have zuranolone in clinics for treatment of postpartum depression. The timing, so we've used zuranolone in the experiments, and we again see with our protocol, our 15-minute protocol, we see zuranolone increase the current density of tonic current. The treatment is 50 milligrams once a day taken for 14 straight days. And so we asked the question, does this constant treatment of steroid affect GABA receptor distribution? And this is some preliminary data that we have. Abby was just analyzing more data as I was leaving for the conference. But again, we've looked at mass spec readouts from alpha-1 containing receptors and alpha-4 containing receptors after 5 days of chow, 516 containing chow. And so the mice are constantly consuming the neurosteroid over 5 days. And so what we found was that after 5 days, we start seeing, in the alpha-1 containing receptors, we start seeing more alpha-1 come through and more contributions by other alpha subunits. And we also see increases in beta-2, but a drop in beta-3, and we also see significant contributions by gamma-2. The alpha-4, on the other hand, is we see some alpha-1 containing receptors. Obviously, we see alpha-4 because we've pulled from alpha-4. We've seen a slight increase in beta-3, and now we're starting to see delta. This was just preliminary data. We've added more N numbers now. This is now significant change. So the subunit composition has changed with drug exposure. So the summary: neurosteroids show a lot of therapeutic potential. They allosterically bind and modulate GABA receptors, but they can also bind to these G protein coupled receptors and enhance tonic current through a metabotropic pathway. And we believe that membrane progesterone receptors could be a potential new therapeutic target to enhance tonic inhibition without so ORG that I mentioned, that's a non-specific NPR agonist, but it doesn't have any allosteric modulation on GABA receptors. And so one of the issues with treatment with a GABAergic allosteric modulator is that there's a lot of sedation that occurs using those drugs. And so by enhancing tonic inhibition that's not GABA allosteric potentiation, we believe we might avoid the sedation side effects. So with that, I want to thank everyone in the lab and our funders, NIH, and Sage Therapeutics. Thank you for listening.
All right, do we have any questions?
So Paul, it's really nice talk. So I'm really curious about the alpha-4 containing subunits in the endogenous mouse brain. You only show that alpha-4 can associate with beta-3, but not gamma-2 or maybe delta. So do you have some explanation about this, or is this the same case for alpha-6 containing subunits? So what's the potential role of gamma-2 in the actual synaptic site?
Yeah, we didn't look at alpha-6. When we had that data, it was kind of surprising based on current dogma. But what was even more surprising to us was when we looked back in the literature, way back in the 1990s, there is some data out there that suggests alpha-4 and beta-3 are the main alpha-4 and alpha-4 and beta are the main composition of alpha-4 containing receptors. With early delta anti-sera experiments from Hans Mohler's group in the 1990s, they published that only, I think it was like 8% of alpha-4 containing receptors are matched with delta, which was kind of surprising. So we're not saying that it never exists. So the thing with mass spec is that we only go with the data that we see in every replicate of the experiments. So we do have replicates where alpha-4 is with delta, but then that doesn't come through every replication that we do with the experiments. We just focus on the ones that are the most common. So we're not saying that it never exists. It's not the most common form of a receptor, but it does change with drug exposure. We believe that there's alpha-4 beta delta in ER waiting to be signaled to come to the surface of the membrane, and with phosphorylation, that's a signal for it to pop in and to have a different property, because alpha-4 beta-3 delta, and especially when beta-3 is phosphorylated, those receptor subtypes are spontaneously open. So you don't need any GABA to open the channel. They've open and conducting. And for mice that have been treated for 5 days with SAGE, we have electrophysiology evidence that the tonic current is spontaneously open.
Yeah, that's really interesting. Yeah. So my question is follow-up. So do you think that is region specific? In thalamus, you will see alpha-4 beta-3, but in cerebellum, you will see.
Oh, we didn't look in cerebellum.
So, yeah, you may see.
We in cerebellum, you might see alpha-6 beta-delta for sure.
Yeah. So, about subcellular location, do you think alpha-4 beta-3 is mainly in a parasynaptic or extrasynaptic instead of a synapse?
Yeah, so for mass spec data, I didn't show, but we pulled, I'm blanking on the numbers now, but I think we pulled
Everything.
660 to like 80 different proteins, synaptic cytoskeleton proteins with the alpha-1. No cytoskeletal proteins came down with the alpha-4 subunit, and so showing that alpha-1 is really synaptic.
Yeah.
And alpha-4 is not synaptic. It's not associated with any synaptic markers at all.
Yeah, I think probably what you see, alpha-4 beta-3 is more like an extrasynaptic.
Yes.
Alpha-1 beta-3 gamma-2 is more synaptic.
Exactly. Yeah.
Sorry, Paul, I'm going to ask a very obscure technical question.
Okay.
So when you do patching,
Yes.
yeah, and you get channel rundown over a period of time, particularly with GABA receptors. And you always ask, what is that on about? Are you telling me that actually that's just, to some extent, the neurosteroids leaving the membrane,
Yes.
causing channel
Could be,
membrane?
yeah.
Is
yeah.
that?
Could be.
Could be.
No, so
yeah.
I smile because when you say rundown, that was the first electrophysiological test that we did with this study. And so when we stimulate PKC in a HEK cell containing alpha-4 beta-3, we don't see the rundown. It's maintained within in the membrane. But without just vehicle controls, you get rundown. And presumably, and I don't think HEK cells have endogenous neurosteroids. I don't know, but you've got this constant recycling of receptors, and eventually it just like runs down, and the receptors are just being removed from the membrane, and they get less and less and less and less with whole cell because you're washing out the kinases.
That's right.
And the signaling, but like if you then stimulate PKC by either adding a PKC activator or adding the neurosteroid, and we added it to the inside of a patch electrode as well, so it was just going in. So when we did that, it just kept the PKC going, turning over, and phosphorylation.
I'd also say it's like, it's one of those differences that we talk about, but we don't really think about with the EC50s between oocytes and HEK cells, and may explain that difference. The other technical question is, so you're showing quite clearly there that alpha-4 beta-3 receptors form together and alpha-1 beta-3 delta receptors. So could you type that out into a letter and sign it so we can send it to reviewers, because it's much easier to record alpha-1 beta-3 or alpha-4 beta-3 receptors rather than alpha-4 beta-3 delta receptors.
We believe me, when we've published this data, we've had to. It's one of the reasons why I mentioned the 1990 papers, because the data's there, and so between then and now, something happened, and everyone jumped on board of alpha-4 beta delta is and the reason why I showed the interneurons and the biphasicness of that. So the main story that I can tell in relationship to alpha-4 beta delta story is that people did it in slices, and they didn't add GABA to the recording solution. And so when you're recording from a brain slice, you're recording from a chunk of dead brain tissue, right? So, and it's all being washed away. There's no constant activity to increase ambient GABA in like what you would find in a real brain, right? And so if you've got this biphasicness of alpha-1 beta delta receptors on the interneurons, you're going to always have low conductance at those receptors, and so therefore it's going to be excitatory, a lot of GABA release. And then when you have a delta knockout, so more GABA release, more postsynaptic, extrasynaptic ambient GABA. And then when you knock out the delta, in the delta knockout mice, then you get rid of those, or reduce the effects of those extrasynaptic alpha-1 beta delta receptors on interneurons. And therefore you're not getting that increase in GABA release from interneurons. You're reducing the ambient GABA, and therefore you get a smaller tonic current being recorded from granule cells. That's the way I'm interpreting the delta knockout mice data, and the confusion of delta contributing to extrasynaptic receptors.
All right, go big picture again. I mean, one of the remarkable things that was presented by Rico was that across the different subtypes, there was an amazing convergence in terms of phenotypes, right? In terms of the stratification. And to my mind, that suggests there's convergence at some cellular level. What you're presenting is quite diverse expression patterns of these different subunits. And I guess, how do you reconcile what is quite clear clinical data with what is quite complex expression patterns. And again, I guess that leads to the next part of this question, is there a potential converging method to improve things here? Is there a drug that can converge across everything? It's a big question, but it's an interesting finding.
I'm guessing that you come down to circuitry within the brain, and if you might only need to affect one of the subunits, a small amount, to have a global effect, whether it's alphas or betas or gammas. You're going to change expression and function. And so what that overall means is at the end of the day, you're going to have a common disorder. Yeah. Yeah.
But it is, I mean, I would not have predicted stratification to be as consistent across the different subunits.
Right.
On the back of the fact that we know that it's so complex, it doesn't make intuitive sense.
No.
The data's there.
Yeah.
Okay, for the sake of time, we're going to move on to the next section.